Well, the next speaker is Claudia Pérez Barrancos. It's a great honor to introduce you. Yeah. So Claudia is a predoctoral researcher and technician in the Biogeochemical Laboratory at the Spanish Institute of Oceanography in the Canary Island Oceanographic Center. Her research focuses on understanding how marine microorganisms respond to natural ocean fertilization processes, such as nutrient input from dust deposition, hydrothermal fluids associated with submarine volcanoes. These phenomena have a direct impact on marine productivity and are closely related to the global climate. Her work is primarily carried out in the Macaronesian region, where she combines field studies with advanced sequencing and bioinformatics analysis techniques to gain a deeper understanding of marine microbial ecology. She's also currently working on the isolation of marine microorganisms to identify biomolecules with a potential application in biotechnology and biomedicine with her Nature X project framework. So her talk is unveiling the hidden microbial diversity of the Tagoro volcano and its biotechnological potential.
Thank you, Juan Pablo, for the introduction. Now I'm nervous. But well, today I'm going to talk about the hidden microbial diversity of the Tagoro volcano. We've been talking about Tagoro all morning, probably, so I'm going to be really brief with the physicochemical anomalies. Tagoro erupted in October 2011. It has this aspect, main cone at 89 meters below sea level, a main crater at 127 meters depth, and then it continues with at south with some secondary cones. The eruption promoted several perturbations in the physicochemical properties of the water column. But then in March 2012, Tagoro entered an active degassing phase, which involves the release of hydrothermal fluids with warm waters with metals, nutrients, and gases. A recent article led by Juan Pablo determined that the current hydrothermal field extends from the top of the summit of the Tagoro volcano to the main crater at 127 meters below sea level. And it comprises around 7,600 square meters, so it's a big space that it's composed by several or thousands of irregularly dispersed hydrothermal vents that have different morphologies, as we can see here in this photo. We have the typical chimneys, the columnar ones, and then fractures and fissures in the rocks covered with microbial mats. So the water column around the Tagoro submarine volcano still has significant variations in temperature, salinity, pH, and CO2 values, but oxygen profiles are showing that there are not anomalies in this current degassing phase. So also what we can see is that the seawater around the main cone of Tagoro, or this hydrothermal field system, is enriched with inorganic nutrients, and in fact we can see that silicates are a tracer of hydrothermal activity in the area. Here I wanted to put this map just to show us the big importance or the global relevance that has Tagoro, because in yellow points, I don't know if you can see them really well, there are only 3, because Tagoro is one of the 3 shallow intraplate hydrothermal systems identified around the world. So every study that we conduct in this system is a big opportunity to study rare volcanic activity or submarine volcanic activity. So now, what we actually know about marine microbes at Tagoro. Earlier this year, we published a study where we demonstrated via experimental evidence that the fluids that came from Tagoro reach surface waters where there is sunlight, and therefore promotes the growth of phytoplanktonic or photosynthetic organisms. And this supports the whole biological system. This was really important because there was a discussion in this topic for many years. I will be really brief, just to show us some graphs. Here's what we can see is that there was an increase in phytoplankton biomass in those bottles that were affected by hydrothermal fluids. The response was faster and stronger, sorry, here. Faster and stronger in the colored ones than the controls. Then we also observed that the main benefiters from these hydrothermal fluids were small diatoms and small microalgae, but also dinoflagellates. And here I wanted to point out something really interesting that we found, is that there were many parasitic organisms. This was observed before in deep hydrothermal fields, but never in shallow hydrothermal fields, so this was really interesting. And just to show you an example, we saw this dinoflagellate, Blastodinium species, which live in the intestine of copepods. And these allow us to link our observations on marine microbes with previous studies on zooplankton, where they observe a decrease in the diversity of copepods around Tagoro. So marine microbes give us really important information to have the whole picture of what happens in an ecosystem. So now that we studied the epipelagic waters, we were really interested in studying the benthic habitats. This year, I hope at the end of this year we will have published this study. We analyzed 37 microbial samples, this is a lot, from 6 oceanographic cruises between April 2017 and February 2022. We collected these microbial samples around the hydrothermal field, not only in just on one point. We were distributed along all the hydrothermal field to have a really good picture of what was happening there in terms of microbial communities. And we used several oceanographic instruments to collect our samples, also to check if we were having any differences related to the oceanographic instrument that we were using. And we used Niskin bottles, typical rosette sampler, and then 3 types of instrumentation attached to the ROV. And now I'm going to show just the prokaryotic community, because I don't have that time to talk about eukaryotic ones, but we also have the data. Here, one of the main results is that what we can see is that richness and diversity of prokaryotic species decrease while we are increasing our distance to the benthic sources, to the vent sources, because we use the type of sample as a proxy of habitat. So we have here the reference water. I think that Sarah mentioned about a station that we have really far away of Tagoro and unaffected of hydrothermal fluids. Then we have enriched seawater. These are hydrothermal fluids from Tagoro. Microbial mats, rocks, sediments, and chimneys. These give us, therefore, a gradient of hydrothermal activity. Here, don't panic. I would just want to show the importance of using sequencing techniques, because this was a study where all data was from amplicon sequencing. And these graphic bars, what give us is an idea of what microbes are living in that sample. And just to show an example, The yellow, the orange points are the sediment samples, and what we can see is that they are grouped together, and they have this characteristic green prokaryotes, which are thermodesulfovibrionia, and these are strictly anaerobic sulfate-reducing bacteria, and these give us a hint that sulfate was maybe there in Tagoro. And this was aimed to summarize a little bit the previous graph. Here, what I only show is that there are 4 most abundant classes in around Tagoro. And these are the families and orders more abundant within these classes. What we can see here is that, for example, in microbial mats and rocks, we have this abundance of mariprofundales that are ion-oxidizing bacteria. Then we have in rock, sediment, and chimney methylococcales, which are methane-oxidizing bacteria. And for example, the last one, Nitrosopomilaceae, which are important in enriched seawater, which are ammonia-oxidizing archaea. These give us a hint of previously recognized energy sources in the area, like, for example, methane and ammonium. So we need to take a look to this, and we have lots of work to do. Another thing really cool that we discovered at Tagoro is the presence in the chimney samples of this kind of twisted filaments. At first, we didn't know what was that, because scanning electron microscopy can be really fascinating. You see lots of things, but these, because of the chemical characteristics, we know that they are iron minerals that are linked to mariprofundus, which are the previous one, bacteria that I was talking about, that use these twisted, promote these twisted stalks to have like motility, so they can situate themselves in ion and oxygen gradients where they are living and thriving. So this is also a hint or an evidence that there are active bacterial biomineralization in the area. Okay, so now I'm going to try to explain this because it's a little bit difficult. So we have lots of sequences in our data set. And we were surprised because almost 2/3 of the sequences weren't identified at genus level. And at the beginning, we thought, okay, we make something bad at the laboratory, we have a big error, and we don't know what's happening here. So we checked these sequences against global data sets that have sequences from a lot of environments, not only marine environments, and around the world. And what we obtained is that almost all sequences were found in other places. So we were not wrong with these sequences. Instead, are organisms that haven't been able to be identified previously. So this is a really novel observation that Tagoro holds a great microbial niche waiting to be discovered, because we need to take more sampling efforts, and in this case, we need to do culture techniques to try to identify and characterize these organisms. But we are talking about 2/3 of the sequences of a data set, so it's enormous. And an example of this is Thiolaba veneris, which was a bacterium that was discovered at Tagoro. And also, and just to finish, because I don't have many time, bacteria also has the functionality or the characteristic of metabolizing secondary metabolites that has a value for biotechnological or biomedical applications. So there are currently researchers at Tagoro interested in this topic. In fact, we have this article that brings the first data set of bacteria cultured at Tagoro. And they have seen that many of them has these anti-proliferative properties that can be used afterwards in biomedicine techniques. So now we are really interested in this topic. We started earlier this year a collaboration with some of the researchers from the previous article. And in just, I think, 4-5 months, we've got like 100 culture bacteria from Tagoro, and among all of them, I just show one, Alloalcaniroborax, which is an example of this. What I was saying is that this bacteria is capable of degrading hydrocarbons. So for example, we could use this bacteria in bioremediation applications. So it's just an example of what we can do at Tagoro in terms of biotechnological advancement. And just to finish, some key findings or summary, shallow hydrothermal emissions from Tagoro enhance surface primary production and sustain distinct microbial communities. Nutrient-rich inputs stimulate phytoplankton growth. Hydrothermal activity also promotes heterotrophic and chemotrophic prokaryotes. And despite reducing diversity near vents, key taxa driving iron, nitrogen, sulfur, and methane cycling remain widespread in the area. And our results indicate a largely unexplored microbial niche at Tagoro with potential new metabolic functions. So thank you, that's all. Just in time.